Hi, my name is Imani Canton,
My name is Kiera Archer.
Hi, my name is Dina Gibson.
and this is our MSH2 midterm presentation. Here's our abstract laying out the background, methods, and results of the experiment. MSH2 is a member of a set of genes known as mismatch repair genes. MSH2 codes for a protein that plays a primary role in DNA repair. However, mutations in the MSH2 gene can occur and cause Lynch syndrome, also known as hereditary non-polyposis colorectal cancer. MSH2 gene mutations can cause production of an inactive MSH2 protein and thus interfere with DNA repair. When MSH2 is disturbed, the amount of mistakes in DNA left unrepaired will accumulate and cause the formation of a tumor. The goal of this project is to determine whether or not repair is the same or worse than wild-type MSH2. Human SNP is engineered into yeast MSH2 gene, introduce engineered plasmid DNA into E. coli because it produces more plasmid DNA for experimentation. A plasmid mini-prep is carried out to extract DNA. Gel electrophoresis, PCR, and restriction digestion are used to identify the three plasmid DNAs, PMSH2, PMSH2', and PRS413. SP1 and SP4 are the same size DNA, therefore are PMSH2 and PMSH2'. Restriction enzyme BSTY1 is used to cut the MSH2 in order to distinguish between wild-type and mutant MSH2.
Okay, here's our introduction. So now I'm going to talk about our particular project and its relation to Lynch syndrome, or commonly known now currently as hereditary non-polyposis colorectal cancer, or Lynch syndrome. And this particular syndrome is a heterozygous mutation in mismatch pair genes. And the cause of it is by point mutations. And individuals who suffer from Lynch syndrome or NHPCC are at a higher risk of developing colorectal cancers or other aggressive forms of cancers. And those particular cancers include uterine cancer, ovarian cancer, liver cancer, and kidney cancer. Some individuals may have mutations to their mismatch repair genes, and they may not develop cancer. So in those particular individuals, we would call those mutations silent mutations. And for our materials and methods for this particular project, it's various lab experiments that are performed weekly for this overall project. And each weekly lab experiment contains various lab procedures that are related to molecular biology. And some of the lab experiments include plasmid mini prep, restriction digestion of a plasmid DNA, and PCR, or polymerase chain reaction. And the materials that are needed for the experiments within this project, they vary from week to week, but some things are pretty standard as far as using the same plasmid DNA that was extracted from the E. coli strands. And it's also good if you have great pipetting skills, because a lot of the labs that you will do will require pipetting, and your pipetting skills need to be perfect.
The experimental design for week 3, which is our intro experiment, was to isolate plasmid DNAs from 3 E. coli strains using the Zippy Plasmid Mini Prep Kit from the Zymo research. And then we were also required to measure the concentration and quality of our DNA samples using the NanoDrop instrument. The results for that experiment shows that SP0 was 330.5 nanograms per microliter, SP1 was 75.7 nanograms per microliter, and SP4 was 140.6 nanograms per microliter. And their 260 per 280 purity assessment was 1.84 for SP0, 1.88 for SP1, and 1.80 for SP4, which is almost near perfect because the ideal number is 1.9.
This is our experimental design for week 4, which week 4 started our process of PCR and gel electrophoresis. These are just the dilution numbers that we need to dilute it to a 20 microliter reaction, based off of the chart that we just saw with the plasmid DNA concentrations. And this would be for the samples with enzyme, and this would be for samples without enzyme, so undigested samples. We had to just describe what the samples looked like. Basically, they all had the green appearance because of the gel that we need to put in, because of the gel dye, and then that's mainly why they have the green color. And so here's the gel. We did this gel because we wanted to figure out what the uncut and cut versions of the mutant, wild type, and empty vector looked like. Based off this data, we would be able to further our experimentation.
The experimental design for week 5 consisted of us actually carrying out the process of PCR, or polymerase chain reaction, to amplify a region of the MSH2 gene that contains the mutation. And we had to create a master mix for each, totaling up to 15 microliters. And the results from that.
And as you can see from these results, this is our restriction enzyme analysis. So the whole purpose of this analysis was to determine which restrictive enzyme would cut our particular mutant form of the MSH2. And as you can see, we have the wild type versus our mutant, which is SP4. And we determined that the restriction enzyme that will cut our SP4 sample would be BSTY1. And with that particular restriction enzyme, it will cut 13 places within a particular strand of this SP4 sequence.
The reason we did this after week 5 didn't have results because we were just prepping for PCR. And so in order to start week 6, we needed to do the restriction enzyme analysis so we could do the gel electrophoresis. We also did APE analysis.
And we also had to do an APE analysis of the enzyme BSTY1. And as I was saying earlier in this presentation, that it cuts at 13 different sites. And as you can see, if you look on the PowerPoint slide, you can see the 13 different points that it cuts at and the size of the particular base pairs that it will cut from a particular sequence. And on the figure to the right, you see the circular map that is just a summary telling the exact places on the SP4 sequence that it will cut from. And as you can see, our SP4 has over 9,000 base pairs. And so pretty much this is a good restriction enzyme because it cuts away a lot. And if there are any errors within any of those areas, it has a really good chance of cutting away those errors.
The experimental design for week 6 consisted of us actually digesting the 3 PCR products using a restriction enzyme, which we found to be BSTY1, and then to run the digested products on an agar-roche gel to determine which plasmid DNA contains the MSH2 insert. And our enzyme BSTY1, the ideal temperature was 60 degrees Celsius, and the name of the reaction buffer was 10x NAD buffer 2.1. And the bottom chart here just shows the different calculations for our actual mix to digest each different plasmid DNA sample. And this right here is the experimental results for week 6. And as you can see, We have SP1, uncut, cut SP1, uncut SP4, cut SP4, uncut SPO, and cut SPO. The lanes are named incorrectly because SPO and SP4 should be switched. And from these results, we haven't really concluded what the results should be yet, but we'll get to that later on in this presentation.
So in summary and all, the whole point of weeks 3 through 6 was to be able to distinguish between the mutant PMSH2 and the wild type. Because they have the same amount of base pairs, we expected them to be the same amount in the gel electrophoresis, and we expected the empty vector to be a little smaller. Those were expected, and they were based off of figure 1.4, which was our first gel. I can go back to it. This is true. They're about the same size, and this is the empty vector because it comes lower, it's lighter than the mutant and wild type.
Sorry about that. Just give us a moment to fix it.
That was expected. And then in week 6, restriction enzymes were chosen using the NEB cutter. BSTY1 was chosen to cut the plasmid because only the mutant contained this restriction enzyme, and that will be able to let us distinguish between the two. The gel was inconclusive because SP1 and SP4 appeared the same, therefore distinguishment was not possible. SP4 should have contained more cuts based off of NEB cutter because SP4 contained BSTY1, but the wild type did not contain it. In terms of future implications, if we were to redo this, or if we were to, well, after we finish the project, we could look at other genes that cause other types of diseases besides HNPCC. We can look at how MSH2 affects African Americans versus Caucasians or any other ethnicities. We could study other types of genes, DNA repair genes that have mutations that cause other types of diseases. Thank you for listening to our presentation, and I hope you learned a lot about MSH2. Do it. You have to press stop sharing. We have to redo it.